Hello biology apprentices! Today we're going to talk about genetic mutations and some genetic alterations. Hope you enjoy it and let's go to it. The first genetic alteration that we will talk about are the ones that are located in the sexual chromosomes. But what are the sex chromosomes? Remember, these are small fragments of DNA that contains mostly genes that codifies our sex, males or females, but they could also have some special genes that affect body traits. The combination of these sexual chromosomes will determine if an organism is male or female. In the case of humans, the combination XX will be for girls and the combination XY will be for boys. Some of the characteristics that make different the chromosome X from the Y is that the chromosome X is obviously bigger than the Y one. It has at least 1,000 genes and it's going to have just a small homologous fraction with the Y chromosome. On the other hand, the Y chromosome will have less than 100 genes, and as the X chromosome, just a small fraction of it will be homologous. That's why X and Y chromosomes and X chromosomes in the case of the girls will form the last pair of chromosomes in a karyotype because of that little fraction that will be homologous in both cases. Remember that a karyotype is a study that we do generally to know the position and the number of the chromosomes of an organism. From chromosome number 1 to chromosome 44, those could be called autosomes because mostly produce phenotype traits of the body. Finally, the last pair, X and Y chromosomes, or X and X chromosomes, will determine the sex. We are going to focus our attention into the genes that are located inside this sexual chromosome. But let's make something very clear. Because the X chromosome has more genes than the Y, sex-linked mutations in these genes will be more common in the X chromosome. Sex-linked genes, as a definition, will be the genes that are located in these last pair of chromosomes. In the case of humans, in the X chromosomes, boys and girls, we have the genes for the color vision, the availability to see colors. We have the genes for our muscle disposition, the muscle tone, and the general composition of the muscle fibers. There will be also the genes for the proteins that are in charge of clotting the blood. On the other hand, the Y chromosome will have just genes for male characteristics. Now, who will have more chances to have genetic disorders or sex-linked mutations? If you can see this cross, females will have 2 X chromosomes and boys will have just 1. So in this case, the organism that will be most affected for sex-linked mutations will be the boys. Why? Because we only have 1 chromosome X. The other one must be Y for being males. Now, if a gene in the X chromosome is mutated, girls can have another copy of the affected gene in the other chromosome. But in the case of the boys or the males, we only have 1 copy. If we got 1 gene with a mutation and the only copy that we have of the X, we will have the mutation. The first sex-linked mutation that we will see today is known as the hemophilia. Normally, all of us have a protein inside our blood known as 8 factor. It is needed for the normal clotting of the blood, so we don't die by bleeding. But when a person has hemophilia, this 8 factor or this protein will be defective, producing in this case strong bleedings that could lead to death. Let's analyze the genotypes for the person with hemophilia. These are going to be the genotypes for the females. Remember that now we are focusing our attention just in the X chromosome. In the case of hemophilia, the gene will be a recessive one. So for the first girl, we are going to have an homozygous dominant condition, which will lead to a normal phenotype, a lady without hemophilia. In the middle section, we're going to have an heterozygous condition, 1 dominant and 1 recessive. Because I just said that hemophilia is a recessive condition, this person could be a carrier for that gene. Is she going to have hemophilia? No, because she has at least 1 healthy copy in the other chromosome. What will happen with the last example? Because in both copies, that person received a recessive gene, she must have the disease, because both of her copies will be affected. What will happen in the case of the males? If we have this genotype, 1 dominant and a Y, then we are healthy men. But if we had the chance to receive 1 copy of the X from our mothers with the defective gene, we will be hemophilic men. We don't have other place to which we can get this healthy gene from. Y chromosome does not have those genes. The next example for sex-linked diseases are going to work the same. They will be affected genes located in the X chromosome, affecting more males than females. The next one is known as the muscular dystrophy, or also Duchenne muscular dystrophy. It's an alteration on a protein known as dystrophin. This protein normally gives a structure and shape to our muscles, but if we have the gene affected for that protein, our muscles will lack the dystrophin, and then they will be weaker, or they will lack the tone that a healthy person normally has. In the case of albinism, we will have an altered gene for a protein known as thyroxine. Thyroxine is needed for the production of melanin. So if we don't have melanin, all our tissues will be without pigmentation. Sickle cell anemia is another sex-linked disease or sex-linked mutation that affects a gene that produces a blood protein known as the hemoglobin. Hemoglobin is a main component of red blood cells. If this protein is affected, the cell will have a defective shape that will interfere with its natural work, which is carry oxygen around our body. Be sure to understand that this type of anemia has nothing to do with the nutritional pattern that a person can have. Normally, anemia is produced by a lack of iron that affects hemoglobin as well. But in this case, it doesn't matter as much iron consumed in our diet, we will have a defective hemoglobin. The next type of mutation or alteration that we will see today is a non-disjunction. It can happen in sexual chromosomes or in autosomes. What is a non-disjunction? It's when the cell has a mistake in meiosis and their gametes will have more or less genes than they're normally or than they are supposed. In this case, we will have gametes with not just one copy of each chromosome, probably more or less chromosomes than normal. Let's talk about the sexual non-disjunction. Remember that this could happen in both types of chromosomes. Sexual non-disjunction will be when we have more or less sexual chromosomes. The first type of mutation is known as the Turner syndrome. Now, the genotype of this mutation is X0. It is missing a copy of the X chromosome. But because we have all the genes for the female phenotype, they will be all girls because of the only X chromosomes they have. Now, due to the lack of the other copy, they're going to have a short height, they will have neck foldings, they will have learning disabilities, and they're going to be unfertile. The next type of sexual non-disjunction is known as the trisomy of the X. Like the name suggests it, this genotype will have 3 chromosomes X instead of 2. This is the karyotype of a female with trisomy of the X. What will be the phenotype? They will have almost normal phenotype. The only feature will be a certain special feature on the face. They will have a particular face pattern that is quite noticeable, and that will be pretty much all. They can have in some cases some type of learning disability, but that will be all. The next type of sexual non-disjunction is known as Klinefelter syndrome. If you can see here, the genotype will be X, X, and Y. We have an extra X chromosome. What could be the phenotype? Because of the presence of a Y chromosome, we will have a male phenotype, but with mixed sexual characteristics. In other words, it's a male that will have a little bit of breast, a little bit of hip development, but it is going to be unfertile. Probably this person won't notice that has a genetic mutation until he wants to have children, and he may not have because of infertility. The third type of sexual non-disjunction is the Jacob syndrome. This genotype is XYY. We have one extra Y chromosome. Now, because of this extra chromosome, the person will have this phenotype. They're going to be male, obviously, but because of the extra one, we will have tall height, probably learning disabilities, and that will be pretty much all the phenotype. Remember that the actual number of chromosomes in the whole species determines the physical features. So if we have just one extra chromosome, that will be shown in the phenotype. The same will happen if we are lacking chromosomes. The next type of non-disjunction is known as the autosomal or autosomic. Remember that this mistake in meiosis could happen in any of the 46 chromosomes that we have. The previous examples were just happening in sexual chromosomes, but in case of this next syndrome, it will be happening in the chromosome number 21. It is also known as trisomy 21 or Down syndrome. The name is going to explain easily how the genotype will be affected. We will have 3 chromosomes 21 instead of just 2. In this karyotype, we will have 2 chromosomes per type, and in the pair 21, we will have 1 extra chromosome number. Because of that, all the persons with Down syndrome will have kind of the same phenotype, the same shape face, the same tongue that won't fit inside the mouth. They can have more or less degree of learning disabilities. The next type of mutation that we will see is the chromosomal mutations or punctual mutations. Pay attention to the images and then you will get what are these chromosome mutations about. The first type of chromosomal mutation, it will be called as deletion, delete, erase. So we have a normal chromosome here, and he will have the effect of this mutation. If you can see the genes represented with letters, we will have A, B, C, D, E genes in the upper arm of the chromosome, and when the chromosome has the mutation, we are lacking the gene D. Duplication, as the name suggests it will be a section of the chromosome that is duplicated. The genes will be found twice. In the case of this example, we have the fragment B and C duplicated. The next type of chromosomal mutation is known as the inversion. Generally, genes have a particular locus, but if the place is split up or turned upside down, that will be an inversion. Look at the normal chromosome, A, B, C, D, E genes. When we have a chromosomal inversion, the fragment, it's turned upside down. In this case, we have E, D, C, B, E genes. Finally, for the last type of chromosomal alteration, we will have the reciprocal translocation. For this, we need to consider a pair of chromosomes, but they must not be homologous. Remember that the homologous chromosomes belong to the same pair, 1 with 1, 2 and 2, and we go all the way to the 46. But if we consider non-homologous chromosomes, it could be chromosome number 1 with chromosome number 13, for example. They are going to share fragments that were not supposed to be shared. Something similar to the thing that happens with the sharing genes of the crossing over, but in this case, without being homologous chromosomes. All of these genetic alterations, sex-linked mutations, chromosomal non-disjunction in sex chromosomes or in autosomes, and punctual mutations are going to produce changes in the phenotype of the organisms. So it's very important for us to understand how do they work, how can be prevented in some cases, and more important, what could be the phenotype of the organisms that have them. Well, that will be all for this video. I hope you enjoy it. Remember to use your scientific thinking and to love biology. See you later. Bye.